Welcome back to Bio 111. This is Chapter 19, Population Evolution. All right. So a lot of what we're going to be talking about will be population genetics. When we're discussing evolution, we're looking at how the genotypes are shifting in this population. All right, so Section 1 is population evolution. Section 2 will be population genetics. And Section 3 is adaptive evolution. All right, so in the last chapter, we talked a lot about populations evolving and what evolution was, the forces involved. I introduce you to a term here, which is microevolution, and that's the change in allele frequencies. When we talk about microevolution, a good way to remember it is we're really talking about the changes in at the micro level, the molecular structures, the alleles, the actual genes. All right, so we have some mechanisms of evolution: natural selection, genetic drift, gene flow, and non-random mating. Now, we've already seen a few of these terms in the previous chapter, in chapter 18. All right, there's also something called the modern synthesis. We talked about Darwin last chapter. He made a lot of observations. He didn't understand any of the mechanisms, right? He didn't know about genes. He didn't know about much about heritability. He definitely didn't know about chromosomes and certainly didn't know about DNA. If you recall, Watson and Crick and Franklin did not reveal the structure of DNA until the 1950s. So we're talking about 100 years prior to that. But we put Darwin's observations together with modern understanding of DNA and genetics, and we begin to understand a mechanism for evolution in populations. All right, so genetic variation. That is the differences among individuals in composition of their genes. All right, so what are their genotypes? How are they varied? All right, and so what this does, it creates, comically in this photo, it creates different phenotypes. Just the genetic component of a phenotype is influenced by evolution, right? So that's what's being selected against. Remember, when we talk about, this is a bit nuanced, when we talk about evolution, what we're really talking about is the passage of genetic information from parent to offspring. All right? A successful individual, evolutionarily speaking, a successful individual is one that survives long enough to mate, reproduce, and have offspring. All right? That is what success is in evolution. And it is the DNA or the genes that are passed successfully on to the offspring. All right, so some sources of genetic variation. The one that most people are most common with, thanks to things like X-Men and other types of comics, are mutations. And they result in the formation of new alleles. Many are harmful, right? So when we say harmful, we mean because mutations are random, a lot of times what they'll do, they'll destroy the DNA or the gene in such a way that it actually creates either a non-functioning protein or a non-functioning gene, or it creates a gene or a protein that has a new function that isn't good. More likely, it breaks it. Right? We think about breaking some of these check sites in cell growth cycle, the cell cycle, and then we see tumors and cancers being created. Um, Harmful alleles can persist through heterozygote protection. What this basically means, remember we have dominant traits and recessive traits. You can imagine if you have a very rare recessive trait, that recessive trait may never present itself as a phenotype because you have to have two copies of it. And if you never have two parents that both have that rare um, trait, that rare recessive trait, there's very little chance, or there is no chance if you never have two parents that have it, that trait will ever be manifested into a phenotype. This is where inbreeding becomes a problem because you have a brother and a sister that both have this rare allele that is harmful, and they come together, they mate and produce a series of offsprings. Well, we know from our Punnett squares that these sort of heterozygous unions create or have the possibility to have 25% of their offspring manifesting the recessive phenotype. So that's where you start seeing problems with inbreeding. All right, many mutations are neutral; they don't affect anything. All right, maybe they hit a piece of DNA that really doesn't code for anything. Maybe they hit a recessive allele, and well, because it's recessive, it's not doing anything. So, um, it gets masked by this heterozygote protection. Um, it could also be that that you actually have an exchange on codons sequence, but because there is some degeneracy, remember, multiple codons code for the same amino acid. Because there's that degeneracy, it may be that it actually doesn't change the protein, the polypeptide chain sequence. So we end up with no real change. And then some rare ones are actually beneficial, and this is where we get our source of new traits that can be selected against and potentially produce new organisms or organisms with new and beneficial traits. All right. Chromosomal rearrangements or gene duplications. You may recall when we talked about meiosis that during meiosis you have some crossover events where we begin to moving genes around the chromosomes. This happens in some larger extent in plants, where we have these transposable elements that move around the chromosomes. In plants, we also have polyploidism, and this is where in some situations we end up with a plant that has multiple chromosome copies. And what this actually does, if you remember, there are genes on these chromosomes, and this gives a backup gene, and it allows that plant to get a potentially harmful or knockout or a modification to a gene that is needed and not die. All right, so they end up with a modified gene plus the working gene, and they're able to move forward with that and be selected against and maybe move on to pass that new gene to another generation that could potentially see more modification. Gene duplication without polyploidism is also possible, where you actually have a duplication of a particular gene. This can be beneficial. Can also be problematic. There's a disease called Huntington's that sees a duplication of a particular gene, and then the larger number of these duplications of that particular gene increases the chance that Huntington's disease is present. But gene duplication can also mean that you just have an upregulation of that particular protein. It can make it more prominent in the cell, in the organism. Rapid reproduction. Evolution occurs quickly in organisms like bacteria with short generation times. So really, rapid reproduction isn't really a source of genetic variation. It's really more or less just a you hit and fast forward on the whole evolutionary timeline, right? You're getting genetic variation from poor copying of DNA during the cell cycle. You're getting large numbers of organisms selected against, exposed to many different types of situations. So that's where we have some source of genetic variation there. And then sexual reproduction, obviously this is where we get to recombine genes from two different individuals and get new combinations of genes. All right, so population genetics, the study of genes and genotypes in a population. We talked about what a population last chapter. It's essentially a group that interacts and exists and reproduces within itself. All right, a gene pool. So that's all the alleles for every gene in a given population. Each member receives its genes from its parents. When an individual reproduces, they contribute to the gene pool. All right. All right, fixed versus polymorphic genes or alleles. If only one allele exists, it is called fixed in the population. In other words, there is nothing. There's an alternative allele that could potentially become more prominent, more popular. All individuals are homozygous for that particular trait. If there are two or more alleles, polymorphic, individuals are homozygous or heterozygous. In this case, you now have the possibility of having some dynamics with this expression of this gene and its occurrence in the populations. Most genes are polymorphic. You hear, we'll talk about allele frequency, or you see it in the chapter mentioning allele frequency. We calculate allele frequency with this equation. It is the number of copies of a specific allele in a population over the total number of all alleles for that gene in a population. So basically the idea is how often does this allele occur in a population over the total number of possible alleles that could occupy that locus on the chromosome. And genotype frequency, that's the number of individuals with a particular genotype in a population over the total number of individuals in a population. So how often do we see a particular genotype in a particular population? All right, so we're talking about frequencies. These are basically probabilities, so they have to sum to 1 when we talk about these. So if P equals allele frequency of CR and Q equals allele frequency of CW, then P plus Q equals 1. All right, one important concept in population genetics or evolution is Hardy-Weinberg stability or equilibrium, and it follows this particular equation. I'm using these carets to represent exponents, And so if a population is not evolving, and we'll get to what we mean by not evolving in a few slides, but it'll follow, the frequencies will follow this equation. So P raised to the second power plus 2 times PQ plus Q raised to the second power, they equal 1. All right, and this gives you the relative allele frequencies in this population. All right, and so think about, allele frequencies. The idea, basically what we're doing is we're selecting alleles at random from a gene pool. How would they, how are they going to come out? So what we're basically saying, we say that there's that this red marble has a 0.8 frequency of 0.8 or a chance of 80%, and the white has 0.2 probability. What we're talking about is if we were pulling these out at random, what are the chances of getting these particular traits? All right. Hardy-Weinberg equilibrium conditions, when we said it's not evolving, what we really mean is that we're not seeing any new mutations in the population. So there are no new variants of alleles. There's no natural selection. So this ecosystem's existing in a situation where there is nothing selecting against anything in the population. Everybody has essentially the same fitness. The population is large, and frequencies do not change due to chance. There's no genetic drift or anything like that. There's no migration. We're not seeing population members move, and all mating is random. Now, these conditions are almost never met, or I don't think they ever are met. No population satisfy Hardy-Weinberg equilibrium conditions. The idea, though, is that it gives us a good basis to compare other populations and to determine if this population, if a given population is actually undergoing evolution. If it differs from Hardy-Weinberg equilibrium, then it's in some form of evolution. In other words, all populations are undergoing some degree of evolution because they're not in Hardy-Weinberg equilibrium. All right, so yes, disequilibrium indicates evolution is occurring. All right, so just a little cartoon showing the difference between micro and macro evolution. So when we talk about micro, like we said earlier, we're looking at changes to allele frequencies, not the size of the organisms. I've got a picture of a bug and a bunch of dinosaurs. We're really talking about, in microevolution, we're looking at allele changes. In macro, we're looking at the long-term paleontology of the change in species, right? Where did the dinosaurs come from? What came before the dinosaurs? Where did humans? All these sort of questions. That's what we mean by macro. But we're talking about micro, the changes in allele frequencies. All right, so how microevolution occurs: new genetic variation. We have mutations. So a mutation can create a new allele in an individual. When this happens, the population experiences a change in its allele frequencies and consequently experiences evolution. So what's going on? We now have new choices for nature to select upon. And nature may find that, hey, this new mutant is better suited. And it outcompetes the other members of its population or its group, and it becomes the dominant allele in the population. That is evolution in a nutshell. All All right. More about mutations. Mutations are random. They're caused by the environment, so radiation, um, some chemicals are carcinogenic, um, or mutagenic rather, um, and that is largely the ultimate source of genetic variation in any population. So natural selection is a blend of chance, so mutations creating genetic variation, or in other words, choices, and sorting, and that's favoring some alleles over others. And that sorting comes from the fitness of those alleles to the environment. And so this third line here is very important. Natural selection is not random. It's not like playing, roulette or anything like this, or flipping a coin. What's going on is that certain traits are better at keeping their host, or their organism alive, right? If you're a rabbit and you live in a very woody area, you're probably not going to live very long if you're bright red, or if you're bright white, or some other color that doesn't appear in your surroundings. In other words, if you stand out to a predator, you'll get eaten. And it's a good chance that you get eaten before you get a chance to, or there is a chance you get eaten before you get a chance to mate. And if that's the case, any of the genes that you have in you, all your DNA is lost to the gene pool, gets taken out. All right, so that's not random, right? The brown rabbits that blend in with the leaves and all the underbrush, they survive, whereas the bright white rabbits that stand out, they get eaten. And so it leads to adaptive evolution, right? You're going to see, our example, you're going to see a shift towards brown rabbits and a loss of the white rabbit gene. All right, so natural selection, it acts on individuals, right? It is the individual rabbit in our previous example that is being consumed, that's being selected against, but it causes evolution of population, because then we begin to see those genes disappearing from the gene pool and not reappearing in subsequent generations, and therefore the population begins to shift towards the brown coloration. Now, if we were to switch the environment and put these rabbits in a snowy climate, say the top of a mountain where there's a lot of snow, the white rabbit would probably be more fit, and we start seeing the brown rabbit decline, right? And in this case, we might actually see, like we talked about in the last chapter, we might actually see speciation, where we start seeing white rabbit species in the mountains and a brown rabbit species in the forest or in the fields. All right, we have this example here of, of these beetles. We have variation in beetles. You have green and brown, and it happens to be that the bird primarily eats the green, or maybe there's um, other selective pressure that changes the abundance of that particular gene in the population. So organisms in a population become better matched to their current environment through natural selection, and this is not intended to mean that the organism, that an individual is changing. What's going on is, if they have a particular adaptation or a particular mutation or gene variation that is beneficial, they survive to reproduce. Those who do not survive to reproduce, and so their genes don't pass on. And it's slowly but surely those beneficial traits begin to accumulate and to build upon each other, until you have some very dramatic changes in the appearance and the survivability of individuals. Differential reproductive success. So individuals with traits that confer survival and reproductive advantage will have more offspring. So you have a fast rabbit gets away from predators, whereas slow rabbits will get eaten. And that means the fast rabbits are more likely to live longer, and they're more likely, if they live longer, they're more likely to produce more offspring or to produce offspring at all. Reproductive success, it can be influenced by a survival advantage, so the individuals live to reproduce, and offer a mating advantage. So we've got an example here of these moths. This is a pretty fascinating example of evolution that we were humans able to see kind of in a short time. So in industrial England, so right at the turn of the 1700s going to the 1800s, England started really industrializing. They used a lot of coal, and they produced a lot of soot. And there's a particular moth that would land and live on these white trees that were around London. Well, as a consequence, because the trees were white, the moths were also white or light colored, and that way they blend in and they weren't eaten by predators. Well, during the Industrial Revolution in England, the trees began to become covered in coal soot and began to look darker or black. And only the moths that had slightly darker pigmentation survived predation. And so those alleles were passed on. And after several generations or several decades, we started seeing that the color in these moths shifted from being light colored to being dark colored. So that's a very rapid form of evolution where we actually started seeing these moths changing color. And no, these moths were not covered in soot. That's not why they were changing color. Here is talking about fitness. We're not talking about fitness like Planet Fitness here. We're talking about the ability to pass your genes to the next generation, the ability to survive. Survival of the fittest is survival of those who are able to pass their genes on to the next generation. And that's really what we're really talking about here. We're talking about the passage of genetic information through time. And genes that confer a advantage to its owner is going to be passed on to the next generation. All right, so this kind of this misunderstanding that natural selection leads to the production of a perfect organism, and maybe it's kind of natural to think that because we're talking about constantly selecting and pruning and perfecting and iterating and creating presumably more and more fit organisms. But in reality, what's going on here is that the environment is also changing constantly. You don't just have one organism evolving at any given time. You have multiple organisms. The things that they're preying upon are evolving to evade them. The plants that they're eating are evolving to be eaten less. The environment is changing because of ecological things. Maybe the organisms themselves are producing changes to the environment. They're consuming more oxygen. They're producing more CO2. They're producing compounds that has some sort of effect to the surrounding. Who knows what they're doing, but the environment is always changing. And because of this, the target of what would be considered the perfect organism is always changing. And so that's not what's going on here. They're not creating a perfect organism. They're creating what is now the most fit organism in that particular environment at that particular time. All right, so directional selection, this is where you have individuals at one extreme of a phenotypic range have greater reproductive success. So we might have a example of particular population that maybe light fur is or dark fur is more fit over light fur, and so we begin to see the whole population shift to another end. We talked about this with the rabbits, right? And in a forest, the white rabbits are likely going to be less fit, and so we start seeing the population shift from the white rabbit population to maybe a brown or darker wood color rabbit. We see the reverse if we were in the mountains with white snow. Stabilizing selection, this is where we begin to see phenotypes that are in the middle or in the center part of the distribution beginning to be favored, and so you begin to see this sort of production of more of a bell curve in the population with selective pressure pushing from both sides. And then you have disruptive selection, and this is where you have two populations that are particularly fit, maybe for different reasons. It may be that you have, so in this case we're looking at salmon size. It could be that a larger salmon is better able to swim upstream and mate, and it has a chance of mating that way and winning over female salmon and spawning. But it could also be that smaller salmon, because they are smaller and are able to get upstream with less energy, they're also able to get upstream and mate as well. It happens to be that if you just are in that center-sized salmon, you're not big enough to swim upstream, and you're not small enough for it to be easy for you, so you just aren't able to get upstream, so you're sledded against. So in this case, it might be that we see this bifurcation in male salmon sizes. Right, this example of these four modes, or these three modes of selection: directional, disruptive, and stabilizing. All right, so directional, this is where we see a shift in the mean of the population. Disruptive is where we see a bifurcation, where we're favoring two particular phenotypes, or typically, possibly two extremes, like we saw the salmon. And stabilizing is where we see selective pressure on both sides, and it's narrowing down on a particular, basically it's reinforcing the current population mean. All right, balancing selection. Heterozygote advantage. Balancing selection favors the heterozygote over the homozygote. So what's basically going on here is that you can, like we said earlier, the heterozygote can mask bad alleles, bad recessive alleles, and therefore you have some degeneracy in the genotype versus the phenotype. So it explains the persistence of alleles that are deleterious in homozygous conditions. this acts as sort of a buffer to mutations and some selective, not selective pressure, but to genetic variation. All right, frequency-dependent selection. So rare individuals have a high fitness, and common individuals have lower fitness. Maintain a balanced polymorphism. So an example of prey that exhibits a rare polymorphism, such as color, may not be recognized by predators, and it makes them more likely to survive. So it might be you have a blue cardinal, and because they're blue, maybe they look like something that a predator doesn't want to eat. And so they're not preyed upon. They're not exactly, maybe they're not exactly more fit, and so they don't gain in prominence in the population, but they don't get wiped out. Another example of this in Richmond County, a local example, is that we have a population of white squirrels. These white squirrels are not albino. They just happen to be a mutation, appear to be a mutation of gray squirrel. And we say this because you can see where some of them appear to have crossbred with typical eastern gray squirrels. We have some mixed coloration in some of these squirrels. But anyhow, these white squirrels, for some reason, they seem to survive. And one possible explanation might be that when they're running across the road or standing in the road, that they stand out on the concrete or the asphalt better, and cars don't hit them as often. Right? So it's a kind of interesting idea that some of these squirrels are being positively selected against based off of whether they're going to be hit by a car. Could also be that because they're novel, they're protected by individuals maintaining the garden, that maybe they're fed more and that sort of thing. So there's also some other things going there. That becomes kind of artificial selection at some point. All right, another one is sexual selection, and this is where a mate chooses another mate based off of some sort of phenotypic, some sort of phenotype. Think, peacocks and bright red cardinals and beautiful wood ducks. The males are always much more brilliant than the females because the males are trying to get the attention of the female. So development of secondary sexual characteristics that aid in mate competition. So canines, body size, antlers, plumage. And this leads to sexual dimorphism, so a change in the way that the different sexes look in a single species. A good example of this is, we got a picture of gorillas here that the male and female look very different. Peacocks, if you've ever seen a peacock versus a peahen, they don't even look like they're the same species. They're very different. Intersexual selection, development of secondary sexual characteristics that aid in mate choice. So another example of this is, we talked about peacock, but another example is baboons having differential coloration and other features that signal to the female that this is a fit or a favorable male. And then runaway selection is where you have this female choice leads to exaggeration, development of exaggerated characteristics that may have a survival disadvantage. And if you think about peacocks in the wild, that peacock probably has a pretty poor fitness. You have these giant, picture of it, you have these giant feathers that's got to be signaling to all the predators, hey, look, eat me, right? And not to mention that it takes a lot of energy to make those feathers, and it takes a lot of energy to carry them with you everywhere you go, and you're longer, so a predator can grab you easier. Um, Definitely a disadvantage, but the advantage is you get to mate, and that's required to pass on your genes. So there's this. Um, Now, they're honest signals. When we talk about that, we're talking about some of these traits they may signal through correlation um, that these organisms, these individuals are healthy. So these giant plumages of the peacock may say, hey, look, I'm able to actually produce these giant feathers. Therefore, I have other traits that are beneficial that allow me to get this much energy to eat this much food to protect myself from these what otherwise would be a handicap because I've got these giant feathers. I can carry this much weight. And so the idea is that females choose based on traits because these healthy genes will be passed off to offspring. All right, that's just going back to that. This is just one example of one possible explanation as to um, why these sort of crazy dimorphisms might be somewhat advantageous to the offspring. Genetic drift. This is a change in allele frequency due to random chance. So it could be that a particular individual is removed out of the population just not because they were fit, not because they were unfit, just because a rock fell off a mountain and squashed one of them, or a human came around. Examples a gnat. A human came around and just happened to swat that particular gnat. That gnat's fitness level had nothing to do about it getting swatched to the wrong place at the wrong time. So these things happen, and it might be that if you have a small population, genetic drift is really exaggerated because that one animal gets squashed by that boulder falling off down the hill could represent a large chunk of the gene pool. In a large population, probably not a big chunk in the gene pool. All right, fixation, genetic drift can lead to fixation for one allele for a gene in population. So that's where you have the frequency reaches 100%. So, for instance, you've got a small population and all of the individuals with a particular trait, they fall off the side of a hill, right? Because the rock falls off and they die. Or they just happen to be in the wrong place at the wrong time. And so now the survivors, due to no fitness based off of their genes, just dumb luck, they're the only ones left. And so now we've got a change in our genetic makeup of the population. So if this happens, there's no more genetic variability in the population, and the gene is monomorphic. Genetic drift reduces the genetic variation in the population. All right. So like I said, genetic drift has a greater impact on small populations just because a single individual carries a greater amount of the genetic load or the genetic the total genes of that gene pool. Now, there's something called the founder's effect. So in this example, we have two variations of what appear to be ladybugs, some yellow ladybugs, some red ladybugs, and we take one red ladybug and we put it on an island. Or we take two red ladybugs and we put them on an island and they reproduce. And so we start making a lot of red ladybugs. They're going to appear to have the same genotypes, the same genetics as the founder, the founding father of that population. And because of that, we're going to see genetics population genetics that result from, you know, basically that subset of genetic information that we gave it on that island. And obviously it's going to have fewer alleles because you've only got the alleles from the parent or parents that existed. One example of this in humans are the Amish population. There's a high incidence of polydactylism. This is where you have additional fingers. And this happens in closed populations where you don't have a whole lot of outmarriage, of individuals going out of the close community to find a partner. Now, the founder's effect can occur, you know, we just showed two examples of them kind of being forced culturally or because somebody picked up two ladybugs and put them on an island. But there's also something called the bottleneck effect, which is where you have some sort of catastrophic event in which a population gets cut down and lost at a or gets brought down to a very small population, such that moving forward you have essentially a founder's effect going on. So about 10,000 years ago, it is believed that the cheetah population dwindled down to only about a dozen individuals. So all the cheetahs in the world were nearly extinct. They nearly went extinct except for these 12 individuals. And from those 12 individuals, the population of cheetahs we have today emerged. The reason why we know this is because there's such little genetic variation in these cheetahs that they must have had a catastrophic event that brought them down to that level. And that bottleneck effect reduces, in this case, or as we've shown, this bottleneck effect reduces genetic variation in the population. It's believed that humans also experience a bottleneck effect many, many, many years ago where the population of humans dwindled down to around 100,000. All right, so genetic drift summary. A significant small population. It's less significant in larger populations. It is random. You know, the boulder falling down the mountain, that individual falling off a cliff, that gnat being swatted by a human, or a bug being stepped on in your house, all random events. It can lead to a loss of genetic variation, especially if that individual had a rare genotype, and can cause harmful alleles to become fixed because you've basically reduced the number of individuals in a population, especially if you have a small population. Inbreeding becomes more common, and so now you've got some alleles that otherwise wouldn't have been common becoming more common, or it might be that you destroyed all other examples, all other alleles, and you just have a single allele available, and now it becomes 100% of the population has that particular allele. All right, gene flow or migration. This is sort of the opposite of the founder's effect. This is where you start having the movement of organisms from one population to another population, and with them, they carry genes. So these new individuals mate with individuals in the new population, and we begin to see an increase of, an increase in alleles, and possibly a change in the allele frequencies of the new population, right? And it is essentially the opposite of the founder's effect. You can have bidirectional migration. That's where you have two populations and they're exchanging individuals. It reduces the differences in allele frequencies between the populations. We begin to see them kind of merge as a single population in this case, and it increases genetic variation. Non-random mating occurs when individuals of similar phenotypes are more likely to mate. And this increases homozygotes, increases inbreeding, um, and decreases heterozygotes, right? Because you begin to have fewer examples of crosses that are of different phenotypes and different genotypes, and it creates inbreeding. We mentioned this a few times in previous slides, but the one issue with inbreeding is that you have these rare traits that, because they are recessive, they're never expressed. But if you have two closely related individuals, the possibility that these two rare recessive diseases are become expressed becomes higher, right? So you got two individuals that have the rare allele, this recessive allele, and they mate. You have a 25% chance possibility that they're going to produce an offspring that expresses the phenotype for that trait. And if it's deleterious, that person's going to have some complications and potentially be fatal. So inbreeding depression, inbreeding occurs when population size decreases due to habitat destruction. And it lowers the mean fitness of population if homozygous individuals have lower fitness values. Right, so you begin reducing the population size, and because of that, you have more breeding, and because of that, you have an increase of these, possibly increase of these deleterious phenotypes being produced, producing less fit individuals. All right, so that's it for Chapter 19 and our discussion of evolution. Hope to see you next week, and thank you.